Hello everybody, my name is Alejandro Prieto and thank you for checking out my thesis defense slash lecture. A little bit of a disclaimer about this video is that the first 10 or so minutes is actually just going to be my audio, and that's just because I forgot to press record during my actual thesis defense because I defended it over Zoom. So you'll see a transition to actual video of me giving the presentation or defense in live time. And this recording that I'm doing right now is one that I'm doing a lot later, and I'm just going to edit it in, and hopefully it's as smooth as possible. You'll see a transition. Anyways, without further ado, again, my name is Alejandro Prieto. My advisor is Dr. Roman Zajac, and my thesis defense is on the population characteristics of the marsh mussel Geukensia demissa relative to sea level rise in Connecticut salt marshes. So the first part that I really put into my introduction was the climate change and their effects. And that's because we know about climate change, right, and all of their effects. We know that there's warmer temperatures, erratic precipitation patterns, flooding, seasonal changes, and of course sea level rise, which is going to be the main focus of my project. But all these things have in common are that we see these climatic factors and they cause rapid changes over long periods of time. And these rapid changes can then cause more climactic factors, thus causing a thing called the positive feedback loop. It's very common, especially when we talk about climate change, and these things are interrelated with each other. To provide some more background between climate change is basically we're seeing in this figure here from NOAA is we see the historical climate from 1971 to 2000, and we see the projected climate from 2041 to 2070. And the dark red spots are basically indicating the number of days that are greater than 90 degrees Fahrenheit. And as you can see, compared to the historical climate, what's projected to happen is a lot warmer temperatures, even in spots that you wouldn't normally see those hotter days. To back this up even further, especially with sea level rise concerned to my project, I got this data from NASA. It's from their satellite, and basically it's showing that by 2070, sea level rise will increase another ½ a foot, and by 2120, sea levels will increase more than a foot. So that's the 50-year and that's the 100-year mark. And as you can see at this top right corner over here, you'll see that the millimeters per year is 3.3. And so these calculations over here are only based off that 3.3 rate of change, millimeters per year. And as you can see, historically, it's never really stayed the same. So this is most likely have changed by now, and these figures have also changed. So to talk about the salt marsh and why sea level pertains so much to it, I kind of got this little figure here that gives a good overview of what the salt marsh really is. Basically, they are coastal systems that have a mixture of salt and fresh water. This is called brackish water, and this brackish water is kind of put into these tidal channels, which connect directly to the ocean. In this study, it'll be the Long Island Sound. And this tidal channel creates these tidal mudflats, and then above there we see the beginning of the lower marsh. After that we see low marsh, and then this middle marsh, which has these puddles and such, which I'll discuss later actually. And then we have the upper marsh, and then past that we see plants like Phragmites, especially in this area, that dominate that upper part. And I really chose this figure because it shows how biologically diverse these systems are. You see a bunch of fish, birds. We even have shrimp over here, and including all the different kinds of vegetation that actually inhabit different zones of the marsh, which I will get into a little bit later. So to go off on sea level rise, this is another figure from coastalresilience.org. And basically they're showing the kind of mapping with confidence of places where they, even even with low sea level rise and with no storm, these models still indicate regular flooding of the high marsh portion. So you can relate back to the figure that I just showed earlier, that this higher level can be more frequently flooded, even with no storm and with low sea level rise. This is indicated by this very darker blue in areas like over here where, I mean, this is the Chapage Island on this side. This is actually going to be Trolley Road. And over here, we see that there's a lot of dark blue, even on the high marsh away from the tidal channels. And basically, relating back to the previous slide that I showed with the salt marsh, basically the tidal channels create this, I guess, yin and yang, if you will, of this flooding where during high tide, the marsh floods, and then during low tide, the flooding actually recedes and goes back out into the tidal channel and back out into the whatever body of water it's coming from. Again, in this case, it's going to be the Long Island Sound. So to talk more about sea level rise and its local impact, this data was taken from a paper which studied Blanca Back, which was a marsh very close to Pleasant Point, which is one of the other places that I actually studied at. This is from Zajac et al. In 2017. And over here, he places Spartina patens and short Spartina alterniflora in these dotted lines, and with these lines where Spartina patens are. So a little bit of a background of this is that Spartina patens is usually a upper high marsh kind of plant, whereas short Spartina alterniflora is more of a mid-high marsh kind of area. And so over here in this, when we see this kind of graph, we're seeing that the Spartina alterniflora actually overlaps a lot with the Spartina patens. And that's just because, you know, again, we're seeing these local impacts of sea level rise, and they're getting used to that. So we're seeing places where they're both found together in some of these cases. And this is tide gauge data taken at Pleasant Point in October 2019. As you can see, even in all different areas in the low, mid-high, or upper high, we're seeing this consistency of flooding. Obviously, not all things are exactly alike, but to see this amount of resemblance between each of these zones is pretty cool. And it's showing that it actually is flooded very, very consistently. So to review about what I just talked about, this is just an example of what I've seen. This is at Chapage Island in 2019, another study site that I went to. And basically here is our tidal channel. And this is called that because, again, the high tide and the low tide controls how it's flooded. And then it creates this tidal mudflat. And then it begins the low marsh, which is usually Spartina alterniflora, tall. In this case, it's going to be a little bit of a shorter one. And this is some more pictures that I got from Pleasant Point, another study area that I did. There's low tide and there's high tide. And as you can see, high tide truly is high, and it really floods throughout the whole entire area. And during low tide, that's really where you can step out onto it. And like, for example, during this study, I only had to do this during low tide. I would not be able to have done this during high tide. So during low tide, it is actually low. High tide is actually very flooded. And now to kind of go off of the Atlantic ribbed mussel, Geukensia demissa, which is the subject of what I've studied. So this is a focus on this Atlantic ribbed marsh mussel. It's a species of bivalve filter feeder that is common in low marsh environments. Externally fertilized eggs that the mussels release turn into planktonic veliger larvae, and this larva go into the water column and eventually move onto the marsh with the incoming tide. I place this here because it's important to know that the location really dictates where these mussels can form and where they eventually end up destination-wise. This image on the bottom right corner is an image that shows the mutualistic relationship that this mussel has with spartina alterniflora. It plays a very large role in processing organic matter and nitrogen in salt marsh systems, and they also take in phytoplankton as well. And both of these work together. We usually, what was being seen in past studies, and even now, is that large clumps of Geukensia usually form at the stems of spartina alterniflora. So this is consistent with what we see today. So as I'm formulating my questions for this research, I have three major ones. One was, is the distribution of ribbed mussels expanding onto the high marsh habitats in salt marshes due to sea level rise? Two, what are the population characteristics and dynamics of mussels inhabiting the high marsh? And three, what are the potential long-term population implications for any such expansion? So my methodology is divided into three sections. One, I look at study sites. Two, I talk about field sampling. And then three, I talk about my actual data collection. So the first part are my study sites. So this frame indicates where in Connecticut my study took place. It takes place in two cities in this frame, which is Brantford and Guilford. We have Pleasant Point, which is in Brantford. This frame is showing exactly where my transects were located on this marsh. And then as we go on to Chaffinch Island, we see as well, this is the frame of what we're looking at. My transects were located in these areas. And then finally, we have Trolley Road in Guilford. Also in this frame, my transect would be around here. I'm going to go a little bit more into that, but this is just for you guys to see whereabouts my study took place. So before I actually started to sample, I wanted to kind of decide and look at a little bit more about these different sections of the marsh and explore them further. So to do that, I had to do obviously some literature review, and then I also had to look a lot about the vegetation and what kind of patterns we're seeing on these different kinds of salt marshes. So for example, this is Pleasant Point in Brantford CT. We have the tidal creek, which is down here on the bottom right. And then right next to it is the spartina alterniflora tall form. So this one would actually be, this section that it's pointing out over here would be the low marsh area. And over here, it actually transitions to a spartina alterniflora short form, and then goes to a mix of spartina alterniflora and spartina patents. And then we have the Phragmites australis up top, and then the woody upland all the way up there. And that arrow is just simply showing which direction the tide will be coming in. So after looking at this and a few other LiDAR images of my other sites, I was able to separate the high marsh into three different sections, because that's really where my study is being held, and it's only in the high marsh. So I was able to come up with this kind of coding. So we have our low marsh, our mid-high, our low-high, our mid-high, and then upper-high. So the low-high would consist mostly of a mix of spartina alterniflora, tall and short form. The mid-high would consist of a mix of short form spartina alterniflora with spartina patents. And then that is also containing a lot of pools and pans that are in that area. And then we have the upper high, which would usually be spartina patents and Discicla spicata, depending on the site. And then Phragmites australis, and above that would be the upland. So that would all count as the upland. So I'm going to talk about a little bit about the population characteristics that I measured out there in the field. So the first one is abundance, incredibly important. I am counting the amount of mussels per meter square quadrat. And so obviously this is just a little side note, like when I would make the transects, these transects would be anywhere from about 60 meters to, I think the max was about 113, 120 meters. And I was going every meter square. So that's just the extent of what I sampled. As well as that, I measured size, which are the measured posterior of the mussels, which were protruding from the ground. So the mussels and their mouths would open up on the side of the sediment, usually to breathe and to filter feed. And so I would just measure that amount that was protruding out of the ground. And then I had the number of dead mussels, which are the count of mussels found dead per meter square quadrat. Pretty straightforward. And then I have the elevation, which is the amount of feet above or below sea level measured at the start and end points of each transect. So for that, I actually used a handheld GPS device. So that one as well, I did the beginning and end of each transect to really get a full view of where I started and where I ended and the difference of elevation. I measured the size with a vernier caliper, a dial one, and then all of my observations, my amounts of abundance and size and number dead were all recorded in a notebook. So to go a little bit more into the GIS analysis that I did for part of my results, I first looked at LiDAR data, which was provided by CT Eco. Those images were downloaded as TIFF images, and they were all combined onto one complete raster or raster data set. Once that raster data set was created, I was able to make feature classes, and those feature classes were divided up into two different things. One of them was the actual mussel data, so that would be more of the numerical things, so things like the count, average, size, and death count. And then I had the transects, which were just the line that my data was pointed on. My features were created and labeled, and then all of my field data was added via Excel. So below is an image of an example of what some of the data would look like. I would have the shape as a point file, and then I would have my object ID and then my quadra ID. And then I would just divide into, for example, in this middle column, I had count, average centimeters, which is just the amount that I measured it in, and then the death, the amount of deaths I saw. And then on this column, I had all of my observations. So I was also able to input all that information on GIS. So in that section, I really talked a little bit more about, or in my observations, I mentioned more about the vegetation and the changes from quadrat to quadrat that I saw in my study. So after this, we're going to dive a little bit into my results, which are divided into three sections as well. The first one being my study site descriptions, the second one being statistical analysis of mussel population characteristics, and three, I have a fine scale spatial analysis. So first, I want to go into the study site descriptions. So the next three slides contain images that I actually drew up myself. And then on the bottom right corner, I also have an accompanying image from a drone that shows where exactly my transects were. So this bottom right has all three of my transects here. And then on this schematic drawing, I have exactly what I saw from both observation and the LiDAR data that I collected. So at Pleasant Point, we see that the gradient of the steepness from where the tidal creek began is a little bit steeper, and we have the tall form of Spartina alterniflora in the low marsh section. And then I was able to separate that, the low marsh to the high marsh, and within the high marsh, I created my three different sections. So my low high, my mid high, and my upper high. In my low high section, I saw that I saw mostly a mix of tall and short form Spartina alterniflora and some Spartina patents. In the mid high section of Pleasant Point, I saw the short form of Spartina alterniflora, and on the upper high, I saw a mixture of Spartina patents and Deciculus spicata. On the marsh edge of Pleasant Point, also from the image earlier, we saw that it was Phragmites australis there. Cabbage Island was very different than Pleasant Point, as you can see already the difference in images and schematic drawings. I saw that at Cabbage Island that there is less of a steep gradient. where the marsh starts, and then the low marsh is significantly smaller than that of Pleasant Point. Appropriately, I also saw that on the high marsh there was a very small section of low high and upper high, and it was vastly dominated by a mid-high section. And throughout this entire section, I saw mostly a mix of Spartina patens and Deciculus spicata. In the low marsh / low high, I saw more of a mix of tall and short form Spartina alterniflora, but as you can see from the drawing, it's incredibly small and sparse. And at the end of the transect, we actually get a sandbar, which is also shown in this image down here. So this would be the sandbar, and I drew that as the marsh edge for Chaffinch Island. And finally, Trolley Road was again different than both Pleasant Point and Chaffinch. I will get into this a little bit later, but Trolley Road is really transitioning, at least from observational and LiDAR data that I was able to study, that it's transitioning to mostly a low marsh or low high marsh environment. So appropriately, I labeled this having a little bit larger of a low marsh over here, and then on the high marsh, I actually made a larger section of low high area, and then smaller sections of mid high and upper high. And in the low marsh of Trolley Road, I saw the tall form of Spartina alterniflora. And then in the low high, I saw a mix of both tall and short. And then as we get into the mid high and upper high, there was a mix of Spartina alterniflora and Deciculus spicata. And we have a woody upland as the marsh edge. It is also important to note from this mosaic of Trolley Road on the bottom right that I only was able to do one transect of Trolley Road, and this is mostly because of the density of mussels that I saw and that the environment is transitioning to a low-high environment. So to talk a little bit more about some of the results I saw, like after I entered all my information on the computer, I used NCSS to generate some results. This is these are some graphs. The left is the mean abundance per one standard error of mussels per meter square, and on the right is the mean dead mussels plus one standard error per meter square. And so what we're seeing here, or what I'm seeing here, is that for Pleasant Point, more of the mussels were found in the mid-high section, and it is also the only site that contained mussels on the upper high section. Chaffinch Island obviously had, as you can see, a lot less density of mussels overall. So there was a lot less of a count, and there were no mussels found on the upper high. And it looks at Chaffinch Island that as you progress further into the high marsh, the less mussels that I saw. And then finally at Trolley Road, appropriately, I only have the low-high section because I only made the one transect. So I would only have that low-high there, and it is a lot denser than both Pleasant Point and Chaffinch Island. I'm going to go in a little bit more into those numbers in a little bit. So to further go into that bar graph from earlier, I have some statistical analysis of the difference of number of live and dead mussels of Geukensia among the three high marsh sections, only at Pleasant Point and Chaffinch Island. So the reason I did that was, again, because I only had one section at Trolley Road, and so it was more appropriate to have my three sections of the high marsh be a comparison between Pleasant Point and Chaffinch Island. So over here we can see that the p-values for marsh section and study site is very significant for the abundance of live mussels, whereas the p-value for over here is not significant. It is also important to note that study site and marsh section, that the study site for abundance of live mussels are not really as significant alone, as well as for the number of dead mussels. And then I performed a one-way ANOVA to show all of the Geukensia sampled in the low-high sections of the high marsh across all three study sites. So these are very important because it shows that live mussels and dead mussels actually have a very high significance when we're looking at the comparison of low-high to each individual site. So that was interesting to look at. So I have some results on the population size structure. So this is more about the amounts and the sizes that I took of the actual Geukensia. So these are 4 histograms I created. The one that's labeled A is the number of Geukensia cumulatively at all sites of the high Marsh location. So I have the color codes on the top of each histogram. So I have the low high is blue, mid high is red, and the green is upper high. So at overall, I saw that the size classes of Geukensia were mostly in the size class range of 4 centimeters. And then B over here is the amount that I saw at just Pleasant Point. So at Pleasant Point, I saw that mostly I see a lot of the mussels on the mid-high section, mostly having the size class of 3, 4, and 5 centimeters. That's what the vast amount were. C is Chaffinch Island. So over here, obviously it is a lot less, like I said earlier, a lot less density or count of mussels overall. So it's important to note that the max on the y-axis for Chaffinch is 100. So it is a lot less, and that the amount that I saw again is size class of 3, mostly on the mid-high section. And since Charlie Road, again, I only did the one transect there, I decided to do an analysis more based on quadrat instead of the entire transect. So on the bottom, I have the numbered quadrat position, so 1 through 13, and then I have on the top my codes for the size class. So again, we're seeing a larger density of mussels at Charlie Road per quadrat, and that most of them lie between 4 to 5 and 3, centimeters for size class. You can see that in the beginning, they actually start off smaller and more in the 3 centimeter size class, and then they progress to go to the 4 centimeter size class in the middle. And that stays pretty consistent and then goes back down to about 2. We're seeing 3 and 4 size classes over here. So what I based those histograms off of was my contingency table, which is the table 3 on the left side. So over here, I have all of my frequency of size classes of Geukensia sampled at each study site. So I have Pleasant Point. The max amount of mussels the total amount of mussels that I sampled at that site was 1,329. And then Chaffinch Island, like I said earlier, a lot less. So I only saw 197 at that site alone. And then at Charlie Road, another great difference is that out of the 13 quadrats that I did there, I saw 341 mussels. So in those, an important note is that, at Charlie Road, because it's transitioning, I saw a lot more mussels at that location. And then on the right here, I have a chi-square contingency table that displays the individual sizes of mussels sampled at Pleasant Point, Chaffinch Island, and Charlie Road. So I have my null hypothesis, which are the size classes of Geukensia. Are they independent from where they were found on the high marsh? So, and over here I have that Pleasant Point and Charlie Road were both not independent of those factors. And then Chaffinch Island was the only one that I was not able to reject that HO or null hypothesis. The fine scale spatial analysis was really just of Pleasant Point. I have 3 different maps that I created in ArcGIS. The one on the far left is my Pleasant Point West abundance distribution. The top one over here is the number of dead, and the one on the bottom over here is the Pleasant Point size distribution. So to go a little bit further into this detail, again, I was able to map out the transect and quadrat by each individual quadrat because I went by each meter square quadrat. So I have these transects labeled 1, 2, and 3. Where we're seeing in comparison in the abundance map is that once we get to about 20 or so quadrats, there is a large die-off, so I see no other mussels in that actual transect after 20 or so quadrats. And then in the second one, it actually goes a little bit further, and then I still see a cutoff around here where I see none. And then in 3, the transect was entirely different. When I was sampling, I was kind of expecting to have my work done by about, 60 or so quadrats, but it ended up being about 95 quadrats that I almost saw at least a mussel in each individual one. So that was also interesting to note. On the right side, we see the size distribution, especially in that third transect, that as we start off, the sizes get actually progressively larger as you go to the upper high and mid-high section of the high marsh. And then the number of dead in these transects actually remain consistent for the most part. So a good thing to note is that wherever there are abundance of mussels, wherever there are mussels, there will most likely be dead ones next to them or around them. So that was pretty consistent throughout those transects. So now to go into my discussion, I divided it up into 2 sections. The first part is the comparison to previous studies, and then number 2, I have my potential causes of variation, so why it is different. And as A, I have tidal inundation. For B, I have vegetation, and C, I have recruitment, and D, I have predation. So in a comparison to previous studies, I have my authors on the left side over here. The first one, so as you can see, there's a common name here. It is Mark D. Burtness. He actually did a lot of work with mussels in the 80s, and he is really, a lot of the literature that I reviewed was either based off him or by him. So in his first study in 1984 about this sort of topic, Burtness examined populations of mussels within vegetation types in the low and start of high marsh, meaning that it was mostly a study on the low marsh and small portions of the high marsh. He found that densities of mussels in the short spartan alterniflora zone on the high marsh / mud flat or on the marsh flat were notably lower than those on the low marsh. Mussels in Spartina patens and Deciduous baccata zone were almost absent. And in his second study, the next year, Burtness paired up with Groschholz, and they sampled the first 3 meters of the high marsh, which is analogous to the low-high section that is in my study. And some parts of the high marsh was dominated by Spartina patens. Geukensia abundances were highest in the low marsh area of the Rhode Island salt marsh that they studied at. So in a comparison with my findings, I saw that Geukensia is occupying high marsh habitats at higher densities than previously reported. I know in the Burtness 1984 paper, he did mention some of his actual quadrat by quadrat findings, and they were very different. So overall, the mean density in the low, high, and the mid-high sections ranged from approximately 6 to 10 mussels per meter square, with about 2 individuals per meter square in the upper high section of the high marsh. Again, comparing Burtness and Groschholz's 1985 paper, my results indicate that mussels in almost all size ranges can be found in all sections of the high marsh. So to go with that, in that 1985 study, again, they only measured the first 3 meters of the high marsh, whereas my study was anywhere from 60 meters to about 120, 115 meters by meter square. So a lot more space of the high marsh was covered in my study. However, the population is made up of increasingly greater proportions of larger mussels moving from the low-high section to the upland transition. So next is tidal inundation. Each site was very different, as you can see from the schematic drawings, and as well as tidal inundation patterns. So Pleasant Point has a relatively regular tidal inundation of the high marsh. Mussels here were found throughout the high marsh, and including the upper high section. a good note is that Pleasant Point is the only site that I was able to find mussels on the upper high section. And the maximum densities of those sections was 19, 40, and 15, respectively, of low, mid, high, and upper high. Chaffinch Island, the population characteristics here appear to be most similar to the patterns that Burtness talked about in 1984 and in Burtness 1985 with Groschholz, with few mussels occupying high marsh habitats dominated by Spartina patens and Discipla spicata. But this marsh does not exhibit the types of vegetation changes associated with increasing tidal inundation due to sea level rise, meaning that these vegetation changes may not be related to sea level rise at all, and more of just the way that the marsh is and how different it is from Pleasant Point and Charlie Road. At Charlie Road, it is also important to note that it is transitioning to a low-high marsh environment. I know that's definitely what I saw when I was there, and it is dominated by this intermediate and short form of Spartina alterniflora. And this type of marsh environment appeared to provide a suitable habitat for clusters of mussels to form, in addition to supporting relatively high mussel abundance. So that kind of goes with the earlier data of my graphs and whatnot. So next is the vegetation effects on recruitment. So recruitment to the upper high sections of the high marsh is likely limited by less frequent tidal inundation and the presence of thick patches of Spartina patens and Discipla spicata. On the left here, I actually have a picture of Spartina patens. I know I've mentioned it so much, and I know not everyone may not know what it looks like, but that's really what it looks like. That's really what it looks like. It looks very dense. What I may be speculating is that this could actually prevent recruiting mussels to reach and adhere to the marsh substrate underneath. When I was measuring like this kind of cover, this patch cover, it did cover a large section above that substratum. So that's a very important thing to note. However, once recruited, they may attain large sizes by not being as susceptible to marine predators such as crabs and fish, which depend on tidal inundation to allow them to feed on the high marsh. And then they can also be less visible to avian or mammalian predators. So we're seeing some give or takes with this kind of vegetation type, some pros and cons, but again, worth noting. And then as far as recruitment goes, there are three major points that I really want to stress because they're really noted in past literature, and it's what we know as of today. And that one, most notably occurring at low marsh in clusters. So like I said earlier, clusters of Geukensia are found in the low marsh environment in clusters. Spartina alterniflora presence is common, and then tides ultimately dictate the settlement and location/destination of these mussels. So a picture on the right actually shows a dense cluster of Geukensia at Charlie Road, and these are actually fastened to the stems of Spartina alterniflora. And so to speculate even further, I suggest that as sea level rise increases, mussel recruitment on the high marsh may be provoked because of this, or it may spike because of this increase of sea level rise. And to go furthermore into recruitment, there are some papers like Franz, who studied in the late 90s and 2001, talks about a secondary settlement that Geukensia may actually have. Once they're juveniles, they actually recruit about from 5 millimeters to 10 millimeters in size. They may actually move a second time onto the high marsh. And this is just because of the consistency of inundation that they may see. Another possibility of how they end up on the high marsh is ice rafting. This was mentioned in the Burtness 1984 and the 1985 paper with Groschholz. Ice rafting is when water ice dislodges the marsh sediment, moving the substratum and mussels embedded in them to higher sections of the high marsh. So this one is also a little spotty. It's interesting because although ice rafting may be able to move mussels to the high marsh, not all of them may survive. And this is just because of the change of environment over a short period of time. Not all the mussels are bound to survive that kind of change. The next bullet point I have on here are that mussels may have a high mortality and reduced growth at 15 degrees Celsius. Spawning of mussels would occur between 20 and 23 degrees Celsius, but temperatures can reach 45 degrees Celsius in patch types like pools and pans, that's on the picture on the left and on the bottom there. So these kinds of patch types may not create an environment that the mussels will survive in, because the heat stress is it is definitely eminent in these kinds of patch types. And again, these are pictures from my study on the bottom there. I did see a large number of mussels that did die or were found dead on these kinds of patch types. So, these pools and pans can create temperatures that may not be suitable for mussels in the long term. And then finally, predation. So predation is mostly, it's common that the blue crab is the main predator for the Geukensia mussel, but there's also herring gulls and raccoons that feed on them. So those kinds of predators may feed on newly settled mussels, but would have to travel through dense marsh grasses in order to prey on the mussels found in the high marsh. This means that predation is mostly common in the low marsh environment and not really the high marsh, meaning that as we get into the high marsh, it becomes less of a significant factor. Mosquito ditches were also present at all of these study sites, and these actually may allow predators like marine ones such as fish or crabs to come in further into the high marsh to prey on mussels with the tidal heights increasing. And so in conclusion, in my study, I found that Geukensia are now found in relatively high numbers on the high marsh habitats in several Connecticut salt marshes. With increasing sea level rise, more of the high marsh is regularly inundated and at deeper water depths. These changing conditions appear to allow for more successful larval recruitment of mussels onto the high marsh. And that Geukensia populations sampled in this study exhibit variable population characteristics across each study site, meaning that this creates a suite of complex factors that impact the distribution of mussels, their abundance and size, as well as survivorship and recruitment among different marsh systems. And now I will be taking any questions if anyone has those. Stop sharing my screen here.
So if you all want to unmute and give Alejandro a hand, we can. Yeah, there we go. All right, very good, very good. So okay, thank you for that presentation, Alejandro. What I'd like to do is to open it up to questions, first from the faculty that are present. And so if you'd like to ask a question, please unmute yourself and go ahead and ask Alejandro any question that you may have.
I have a question. I actually have a couple of questions.
Chris,
Sure.
go ahead.
And as Dr. Zajac says, as a budding environmental scientist and ecologist, they may be stupid questions, but I want you to answer with.
There is no question that is stupid.
So what do you mean by recruitment?
So recruitment is the term that is given to mussels that when they do actually spawn, they recruit, meaning that they move onto and settle onto the sections of the high, or whatever marsh is. It just means the movement of and onto the marsh.
Okay. Are these commercially viable mussels, or are these ones that wouldn't be harvested for eating down at your local seafood shop?
So that's actually funny you mentioned that. I actually did. When I actually went to go, I think it was at Chaffinch Island when I got out of my car to actually start sampling. one guy came up to me and asked me what I was doing, and I told him what I was doing. I said I was studying this section of ribbed mussel, and he said that he used to eat them all the time. Now, I don't know if that was just a certain case that is totally separate, but I know that this kind of mussel commercially is not popular to eat. You would normally not eat these mussels.
Okay, is that due to like filter feeders in marsh areas? Is that why they're not commercially viable, or is it they don't grow to the right size, or do you know why they're not commercially viable?
So from what I've read, you know, from things that I've looked at in my free time and stuff like that, it looks like that this species does not reach the size that actually gives a lot of meat on the inside, meat for people to consume. So that's why they're not commercially viable. As well as, you know, being on marsh systems don't really, you know, because we eat clams and oysters all the time, and those are commonly found there as well. But it's mostly the size that people have, that people, I guess, get turned off by this kind of mussel to eat.
Okay, and last question for me. You say that you were using LiDAR imaging.
Yes.
I know of LiDAR imaging from archaeological types of things. Could you explain to us or to me what LiDAR is and why you're using LiDAR images rather than just, say, drone imagery?
Okay, so that's a good question. So to compare drone imagery or UAV imagery is really just snapshots and pictures that you would take from above, I guess, certain elevation. I'm not really sure. I'm not sure what kind of what kind of distance you can get. But number one, LiDAR gives a greater distance, and LiDAR actually uses lasers instead of actual imagery to make images. So LiDAR uses a variety. It usually fires. I remember this one study I read that was actually took place in, I believe it was in Mexico, because they were studying the ancient Mayan civilizations. When they were doing LiDAR imaging there, about 6 billion laser-like pings were fired from a flyby plane. And so as these lasers go down, only a certain number of them bounce back up, and they actually read the direct elevation of each individual laser. So that way it can create a more accurate image of what I'm looking at underneath a certain canopy. So the reason why it was used in the Mayan civilization thing is because obviously the canopy there is incredibly dense. So in order to see underneath that canopy, you would use LiDAR to actually pinpoint small things underneath that and finding something under that. So that's why it's commonly used in archaeology. And the reason why I use it for my study is because the way each marsh system is situated, it makes it hard that when you're on there, you can't really detect any elevation change. But when you look on LiDAR imaging, you would actually see that there is a certain difference. So that's why it's more to catch those small differences of elevation and change from site to site. That's why I used it.
Perfect. Thank you. That's all for me.
Cool.
Okay. Anybody else from the faculty? Okay. Well,
I have a question from Michael Rossi here, actually. So, okay, so Michael Rossi asked me on chat, he said, do you expect that the lower high marsh area is becoming lower marsh at all three different sites over time? So that is a good question. For that, I would more say that Trolley Road, if anything, would become more of a low marsh or low high marsh over time. But at the other sites, it's difficult to say where they might be headed to. Personally, I think that the low marsh can increase over time if sea level and the inundation rates become consistent enough. So meaning that, besides storm surges and stuff, if regular tides frequently inundate the sections of the low high marsh and low marsh, it's very well possible that the low marsh may become larger and may expand. So I hope that answered your question.
Yeah, that's actually a big question currently, just for the sake of discussion, in the sense that, are we going to see a steady progression as sea level rise increases, and at Trolley Road, as you pointed out, Alejandro, is sort of transitioning now from sort of this low high marsh to just low marsh.
Exactly.
And in many sections of that, but yet, you know, down the road at Chaffinch, you know, we see certain kinds of indicators, but they're very different.
Mm-hmm.
And so the question is, more broadly, is how is each marsh system going to respond to these changes in tidal level inundation, and indeed the patterns of that.
Right.
And so, you know, is it going to be the steady progression? But other data suggests that, you know, it's going to inundate and then just totally collapse.
Yes,
so you won't see a steady progression. So, you know, that has important potential management. You know, are we going to see a steady progression of change from high marsh to low marsh, blah, blah, blah? Or is it just going to sort of go boom and then go, okay, no more marsh?
Right.
So in some areas you see indications of that, in other areas you see a progression.
Right.
Well, and then also, sort of to follow up on that, you also have impacts on zonation. We have increased freshwater runoff, right? So that can also bring some of the high marsh down into low marsh. So I guess around these marshes that you were studying, has there been any, you know, changes in development, you know, increase in unpermeable surfaces that might increase that freshwater runoff?
That one is difficult to say whether I personally saw it or not. I know that when there is freshwater runoff, it can mess a lot with the salinity of the water that is running onto the high marsh section. It's difficult to say whether I'm seeing that at some of the sites. I know Pleasant Point is, there is a lot of like pavement around because there's streets nearby. Trolley Road is a lot larger, so maybe it pools in both freshwater and saltwater a lot easier. So maybe that's why we're seeing the change at Trolley Road, for example. And Chaffinch is a little different than both the other sites. It's more of a park, so there isn't much pavement around. So I feel like Chaffinch Island may be more associated with its own devices of vegetation and stuff that maybe show more of a high marsh like area of, you know, in that place. So, but yeah.
And this isn't a question. I just wanted to say I really liked your drawings.
Oh, thank you. Thank you. I'm glad you appreciate that.
Yeah, very nice.
Actually, along of what you were asking, Amy, we had a thesis completed and presented, I think, a year and a half ago, roughly, now by Matt Ormrod. And Matt actually looked at the upland high marsh transition on a whole bunch of marshes in Connecticut. And they are very different. And in some cases, you see this sort of regular, typical zonation from sort of the upper high marsh into the upland transition. In other areas, they're kind of muddy, which suggests the freshwater ground flow input is making it fresher, so you have these fens that are being developed. And then actually in other areas, that upper area, when it gets inundated more frequently, is actually collapsing the sediments and it's ponding salt water. And we see tall Spartina alterniflora at the upland transition.
That's crazy.
Yeah, you can actually see it, not so much at the sites where Alejandro is working, but if you go across the way where you have these rocky headlands and the water has no place to go, it actually ponds and causes it to be more salty. So you have this weird thing where you have tall Spartina alterniflora butting up against the upland transition. And he found that actually on several other sites as well. So it's crazy. I mean, it's
yeah, what's it doing?
Well, exactly. You know, it's you know, what it seems to be doing is that the sediments, because of the increased salinity there, all of a sudden are just sort of just getting really, really muddy and then holding that salt. And so you have this sort of weird, you know, situation as described.
Yeah. I guess I just worry about the compounding effects of sea level rise and also increased urbanization and more of this flow. It just seems like Well, I guess we're already seeing that.
And actually, where they've seen these sort of fresher water fens develop, you can see the vegetation change, but a lot of places it's not where it's urban developed.
Oh, really?
Yeah,
Okay.
it's coming off of forested land and so on and so forth, where groundwater groundwater flow is more. There's more of it. Well, that's what the initial sort of this early data suggests. Yeah. I guess I'll be the first to congratulate you, Mike.
Thank you very much.
No, not you, Alejandro. I'm congratulating on the sidebar. Anyway, I have a couple of. Any other faculty have questions? No? Okay, I have a couple of questions.
Sure.
All right, so these are the typical sort of, hey, so what would you have done differently if you were going to do it again?
If I was going to do it differently, I think I would have gone more with a, I would have done what's called like the bulldozer method of just like collecting as much as possible and then just collect and then trying to figure things out after that, because I think I would have liked to have more vegetation data of each like individual quadrat and like the transition of each one. I did mention it in my notebooks, but I should have like paid more attention to that for sure. And so, because I think when I started the actual project, I was like, oh, like it's on like mussels, like that's what I'm studying, like mussels. And like I'd already done previous work on oysters, so I was pretty familiar with filter feeders. It was just different. But then as the projects kept going forward, I was like, okay, well, I had to know more about geomorphology of the high marsh. And now I had to know more about vegetation and tidal inundation, all these other factors. I mean, I didn't even know ice rafting was a thing until when I actually read those papers.
Good to read the literature, isn't it?
Exactly, right. And that's a totally different, thing that I had no idea was going to be part of what I was going to study. So,
okay.
I also would have looked at observational changes in maybe the winter, just to see if those were consistent. That way I could collect more data, and then maybe some experimental things to put in there as well.
You anticipated my next question.
Okay.
So what would you do next? So now you have this background data. What experiment might you run or one could run in the future? What experiment might provide insight into the patterns that you see?
So a few things. Well, maybe just one solid thing that I've been thinking about is maybe putting off. So this is my thing, is that I wanted to experiment whether if they do survive on the high marsh, like how long will they survive, especially in sections of the mid-high and upper high. And so for that, I think I would section off like maybe a quadrat of mussels. I would tag them, and then I would just come back periodically and just to check on them to see if they lived or died over months of time. And then that would also require me, again, to go in the winter time. Like I would have to look at that as well, because that's also really important. If the ice rafting is like a thing at one of the sites, then I want to know about it, and I want to know, and I want to see the change, and I want to see if that does occur, if the mussels actually survive, or what percentage survives of that, and if they actually successfully stay on the high marsh. So I think definitely like sectioning them off. I was thinking about caging a few of them, or like maybe 20 or so, and then putting them on different sections of the high marsh, seeing again if they successfully survive or not. But the thing with that is that when you take them out of the substratum, I feel like it becomes hard for them to actually survive in general. So that's why I would veer, if I were going to cage, I would put them in the ground and then around and then over, instead of like making a cage and then putting them in there. You know what I mean?
Yeah.
So,
okay. So you could cage off just to see what the, again, mortality rates are from different sources.
yes. Yeah.
And track individuals over time to actually look at survivorship. How about recruitment? Can you think of any recruitment experiments?
For recruitment, I definitely would. I would. So that kind of goes a little bit into how I would change things in
when do they recruit to the marsh?
When?
What time of the year?
They usually recruit in July.
Okay. And
Yeah.
when did you do your studies?
I did mine starting in July to November.
Okay, so right around the time of recruitment.
Right.
Okay. Then so go ahead, recruitment.
So basically that kind of goes with like the first question you asked, like how would I change things? So I feel like when I was in the low Marsh section of Pleasant Point, because especially when I started in July, that's when I did see a lot of smaller, like, 5mm to 10mm size mussels that were,
Recruits.
and so I think, again, taking that bulldozer method approach, I would gladly have collected that information just so I can see if they actually stay there, again, sectioning off a place, or if they move. I guess it's hard to tag those because they're a little smaller, but I would like to see, my goal would be to see if they change from wherever they started off and seeing if they ended up further in or into the high marsh, so on and so forth. But I would definitely like to collect the actual data of the recruited ones, number one, and then number two, maybe having a small section and looking at that kind of abundance or mortality, stuff. So
Okay, final question that I have. Do you think those so prediction, are those high marsh populations going to grow, say on Pleasant Point?
yes, I think so.
Okay. All right, so in 5 years, we're gonna go check it out.
All right, I'll be there.
We'll do at least one transect. Any other questions by anybody?
Yeah, I have one actually.
Mike? Hey, Mike.
Yeah.
Mike.
Great job, by the way.
How are things?
Oh, thank you.
They're good, but I might be there this afternoon, so we could talk about that later.
Okay. Oh,
But
good.
they're good.
So please ask your question.
Yeah. So when you were talking about secondary settlement, I don't know a lot about mussels and what that is, but it sounded like it was kind of like they voluntarily detach and move somewhere. If that's true, what triggers that process, and how might that relate to the species' potential like adaptability to changes in the environment?
Okay, so I think I'm going to answer that question. Okay, so the first part was more about, okay, what is secondary settlement? So to go a little bit more into that, it's pretty much exactly what you described. They willingly move. They detach from wherever they settled on, and they move forward onto the low marsh or on the high marsh section. And then the last part of the question, you said, what was it? It was like how may that contribute to the survivability, or of the mussel?
Yeah.
So I think that they're actually very resilient creatures. Like, I think their adaptability is incredibly high. So if they are moving, like if they, like, first of all, if an animal is doing that kind of secondary settlement, I think it would already imply that their adaptability is great. Like, they have a very high resilience. And what I've seen, some of them that they've, even in the pools and pans that I observed, not all of them were always dead. I did see some on there that were still alive. So it shows that they do have that kind of adaptability to different kinds of environments in that section of the marsh. Now, whether what causes that secondary settlement, I don't think anyone really knows. I mean, the papers that I read about that was by Franz. He did one in 1998, and I believe one in 2001 about both of those things, about the adaptability and the secondary settlement. And it shows that he, doesn't really have any thing to be like, okay, this is why they do that secondary settlement. I think it's just simply because of the tide. They may choose to actually voluntarily move onto the sectors of the high marsh just because of how consistent the tide be. I think that's my personal way to answer that question, because I think when I was reading that kind of literature, he gave all these different things, and what I got from it was that really, like, the tide dictate really where they end up, and Geukensia is very adaptable. Wherever it ends up, it has a very good chance of survival, I think. So I hope that answers your question.
Yeah, it did. Thank you.
Anyone else?
I have one more question that I kind of
Please,
thought of
Amy.
as
Absolutely.
question there, and I'm sorry if you talked about this. I've been getting distracted by emails.
Oh, it's okay.
Trying to focus. Is there any, do you know, is there any sense of how far these things disperse? Like these populations, are they pretty isolated in these individual marshes, or do you have recruitment from neighboring marshes as well?
So personally, what I've been seeing is that I know in the beginning of my presentation, I did mention that they're mostly found on low marsh environments. So that's something that has been established in previous studies, mostly by Mark D. Burtness, the two papers that I mentioned. He's really adamant about stressing that these mussels are mostly on the low marsh environments and very rarely on the high marsh. So as far as I know, I don't mean to, toot my horn or anything, but like this is kind of like the first study of just the high marsh and like their population and dispersal on that, on that area of the marsh. So whether they're together or I'm not sure if you mean by, do you mean like, are they found more in clusters? Because
no, well, just so you know, I see everything in terms of their DNA dispersal. So
okay.
I guess really my question is, has anyone done any like microsatellite work or some mitochondrial DNA so we could actually know some of the connectivity between these different populations?
Yes, so there have been genetic studies, and a lot of the microbiology has been covered in previous papers, but they mostly describe about the the physiological stressors that the mussels can get, mostly from heat and cold, and it also goes more into the way that they process nitrogen and stuff like that. But I think as far as DNA goes and that kind of stuff, I'm not really familiar with anyone that has really done anything on that particularly, and like kind of showing if that dictates their movement or not. I'm not really well versed with that.
Yeah, I guess I'm just curious, like I don't know what we call little mussel babies, do they stay in their own marsh, and like do they get
so if I may, the larvae are dispersed into the water column.
Yes.
Yeah, and where do they go then?
Well, they go out into Long Island Sound.
Okay.
And they have, so they're going to be taken with the outgoing tide, and then they get mixed. And so, but it's an excellent question because we always get surprises in terms of, the extent of larval mixing and therefore the extent of how how similar the populations are genetically across stretches of coastline. And especially for, broadcast spawners, let's call them, such as these. But you never know. But I think there have been some genetic studies that have been done on these guys.
Thanks.
Roman's new nickname is Mussel Baby.
Really?
He's like
really?
You're gonna go there.
I did not give him that
You're gonna go there. God, I can't believe it. Anyhow, anybody else? No? Okay, well then, Alejandro, thank you very, very much. Excellent presentation, and we'll go from there. All righty?
Okay.
Thank you all for attending, and again, thank you very much. All right, take care, and we'll see you all down the line, I am sure. All righty?
Thank you guys for attending.
Do you have any last words, Alejandro?
I have no last words. Just thank you, and hope to see you guys all soon. Yeah, that's pretty much it.
All right, real good.
Thank you for the support.
All right, you bet. All right, folks, later. Enjoy the rest of your day.